My welcome to everybody. So what we're going to try and do is make this interactive. So some of you know each other around the room, but I think it's actually going to be good to get everybody to know each other. So I'm going to try and get people to talk a little bit. It's much more fun when we actually have an interactive session. I want you to ask as many questions as you want. I tried to get everybody to talk a little slower, but also save some time during each session so people can have a chance to interact and really ask questions about what's going on. I want to also thank Terry and Barbara for putting the program together. Without them, all would be lost, that's for sure. So what I'm going to do this morning is quickly go through some of the things that we're going to talk about, but also give you, try and give you a little more. We've been doing this now. I think Charles here. Charles, back in there. So Charles actually started this a long time ago with me. Oops. It does. Okay. And so, and I think this is about. Where's Jane? Jane also. Anyways, and Sasha was here, and there's a lot of people who've been here for a long time. We've been doing this about 10 years now, and we've learned a lot. We're still learning a lot, and so there's sometimes we're not going to give you solid answers to a lot of the stuff that you want to know, but at least we'll tell you what not to do. And I'm going to go into that a little bit more this afternoon. All right, so so the philosophy of the core is really to use metabolomics as a platform for discovery. And all of you obviously are interested in doing this. You know, we do a lot of different kinds of profiling. Oops. I guess that's not the laser. Yeah, I did. Oh, there it is. Okay. Sorry. All right, so we do, obviously we do a lot of different things, and I'll go through some of this, but really what we want to do is utilize the infrastructure that we have here to generate additional knowledge related to biomedical research. Okay. So just a little briefly about how we've set this core up. We were funded as the Michigan Regional Comprehensive Metabolomics Resource Corps. The NIH is the Regional Comprehensive Metabolomics Resource to call it. We're embedded in the Biomedical Research Core facilities, which means that our administrative structure is through the Medical School Administration Office. We report to Steve Kunkel, who's the Senior Associate Dean for Research, and Cassie Wong, who we really report to, is the lead administrator for the BRCF. This does give us access to money and support for a lot of for equipment replacements and things like that. So I get to direct this with Sue Penethor, who's in the back. Sue is a card-carrying metabolomician. Versus myself, who actually sort of pretends to do it. Actually, Steve Brown's no longer a manager. I thought this got changed. Oh, yeah, he's changed it, and I just put my old slides in. Sorry, Terry. Maureen Kochman. Maureen, where are you? So Maureen actually is the person who really makes things happen now. And Grace Wu is our administrator. So is Grace, did you show up? No. She's otherwise indisposed. So we have analytical core, a data core, statistics and bioinformatics core, and a promotion outreach core. This is a program is supported by the promotion outreach core. So all these people that work in the cores really have important roles, and I would urge you to talk to the people and really get to know them, because they can be very important, especially those people that are inside the university. So we've set up the core that it's all run through a lab information management system, a MetLIMS that was started actually when Chris Beecher was here. many years ago. We've actually been using this a lot to handle the flow of information into and out of the core. So some of you have submitted samples to our core. You know that we're obliged to have you submit it through our lab information management system with metadata and information about the data. One of the important things that you'll hear about at some point during the week is the requirement that the NIH has that all the data that we collect on your samples gets uploaded into a central biorepository that's at the University of California in San Diego, such that after you publish your data, that just like transcriptomic data, the data will be released to the public and the data can be mined. I think it's actually really important to do these kinds of things because it allows other people to learn from your data, but also allows you to interact with other people who may have other ideas on how to use your data. It's basically a requirement that if somebody's going to use your data post hoc, that they contact you. There's only a little bit of metadata that actually gets uploaded, and so it actually gives you an opportunity to work with other people to use the information that you've generated so other people can get new knowledge out of it. So it's one of the things, if you come to our core, that it's one of the requirements that we have that your data is going to be uploaded at some point. Some of the journals are beginning to have this as a requirement. So we'll go through that a little bit. I think we're having a video conference, right, from wherever Owen is vacationing. Excuse me. So the offerings of our core right now, so we have a wide variety of things that we can assay. And if your favorite metabolite is not on this list, we can set up an assay for it. We do everything in a fee-for-service manner, such that we can recoup the cost of it. It's an expensive infrastructure, and so we're trying to keep it the costs in a reasonable range, such that at least we're competitive with others around the country. So we can do a lot of targeted metabolomics, fatty acids, trans fatty acids. We've run thousands of trans fatty acids samples. I think Arun Das is here. He's been doing that. We can do acylcarnitines, ceramides, steroid profiling. This is a nice platform that Rich Aukus bought to us when he moved here from the University of Texas Southwestern. Rob Kromick is running this. We do a lot of phospholipids, and you can see the numbers, and these numbers actually are bigger than we keep adding things to these lists. So that's a lipidomics, the targeted platform. You can see we can do a lot of other things as well. One of the most popular things that we do these days is fluxomics experiments. We're using heavy isotopes to trace compounds through different pathways, both in tissue culture, but we're more and more doing it in vivo, looking at how you can see how molecules are distributed throughout the different tissues and how they're metabolized specifically within tissues. And then the untargeted metabolomics lab will give you both a broad array of metabolites that you can measure in a semi-quantitative manner and try and look at the broader changes in metabolism. And finally, we have a shotgun lipidomics platform, which you'll hear about from TM, who developed it with Tanu Soni. That's really a very beautiful platform, and you'll see some of that. So I'll just go through very quickly some of the ways that we go about doing it, and I think most people do it this way. In general, we do mostly mass spectroscopy. We do have a partnership with pharmacology if we want to do some NMR, but most of our work that we do here, probably 99.9% of it actually is mass spectroscopy. We have partners, the other RCMRCs, the 5 other RCMRCs have a little very variable capacity to do NMR, and if we believe that your project is better done at one of these other sites, we'll recommend that you go there for the kind of work. So in general, the workflow for target metabolomics, and you'll hear more about this, is you know what you want to measure, and so you prepare the sample in a manner that you want to prepare, or you should prepare for that specific metabolite or group of metabolites. And then you can put it into instruments, and you can find the peaks that you're looking for, and you can be very quantitative about the amount that's in there by generating calibration curves and quantitating it. And then you can do data interpretation, statistics, and it's pretty straightforward, although it gets complicated when you get lots of groups and lots of time courses and things like that. But this is what we're working on, trying to actually get better ways, especially when we do fluxomics experiments, to try and understand how much is going where. And we'll go through some of that. I think Mahmoud El-Zuni, I don't know if Mahmoud's here, will be giving that lecture. Now I'll just show you a couple examples of how we've done work for both for people internally, but also outside. This is a paper that was published last year with Sharon Wardlaw, taking samples from individuals undergoing sequential sampling of their cerebral spinal fluid. This is a study that we found out about, is that Kevin Bates at the University of at Washington University was looking at the dynamics of beta amyloid protein and infused C13 leucine into people to label beta amyloid and then stop the infusion after 18 hours and then sample for another 24 hours. So we actually talked to Bates, and Dr. Bates was kind enough to give us the leftover CSF. And so we developed a sensitive assay to look at different metabolites. And these are the amino acid metabolites from the plasma and the cerebral spinal fluid that was sequentially sampled. And so you think about the work that went into this to take, and this was just 2 individuals out of the 12 that they did. And so we ran all the samples that we could get our hands on. And what we found is that there's actually a high correlation between the amount of amino acids that are in the blood at any given time. And so if you see here, this is at 1 meal, 2 meals. You can see that there's an up and down in the plasma, and there's actually a nice correlation between the amount in the plasma and the amount in the cerebral spinal fluid. And further, what Sharon did is she looked at the levels of AGRP and POMC and leptin in CSF in relationship to this. And what it came up with is that there's a very high correlation between the POMC levels in the CSF and the leucine levels in the CSF. And this was fairly interesting because several years ago, Randy Seeley, who's joined the faculty here, showed that if you infuse leucine into the CSF of mice, it actually stops them from eating through presumably the activation of mTOR. At least that's what the experiments showed. And so what we see is that POMC, which is an anorexic hormone, actually looks like it's released in conjunction with the rise and fall of specific nutrients, namely branched-chain amino acids. And we sort of like this because branched-chain amino acids are essential amino acids. And if you really wanted to detect your nutritive level in your blood, it'd be actually nice to have something that was that's something you couldn't make. And so we're following up on this with some other experiments. This is a study by Vince Young and Casey Thero. Casey's actually going to talk to you a little bit more about this, but this is just a simple a plot of some of the bile acids that we can do, we can measure. We now have, I think, nearly 30 bile acids that we can measure in the plasma and also in the stool. And what Casey is very interested in is the growth of C. difficile toxin, and C. difficile and the production of the toxin. And what she did is she ablated most of the bacteria in the gut by using antibiotics. And what you can see is that these specific bile acids are expressed in the contents of the let's see, where is it? It's hard to see it from this angle. And so you can actually see it in the upper gut, in the lower gut, and then in the cecum, and then in the fecal material, you can see the different concentrations of bile acids and different kinds of bile acids as you go up and down the gut. When you actually destroy the bacteria, what you see is you only get primary bile acids, and most of the secondary bile acids are gone. And what Casey has suggested is these primary bile acids are specifically involved in the germination of C. difficile. And so one of the upshots of this experiment is that by putting in antibiotics, you destroy the normal bile acid flora, and the bile acids that are left are actually responsible for increasing the germination of the C. difficile. We also have an untargeted platform that you'll hear about. I think Maureen is talking about this. And this is a much more of a semi-quantitative methods where we have a standard preparation for most of our platforms and run it on either LC or GC, and now we actually have a normal phase that we're developing. And what you get is a bunch of peaks, and then through recursive processing, you can align the peaks, get relative quantification of the peak area, go back and look at things and try and line up peaks better. And then what you get out of this is actually a number of different metabolites. We can name about 300 of them in a reasonably reproducible manner and get up to 2,000 to 3,000 other peaks that will be semi-quantitative that you won't know what they are, but we're trying to develop things, and Ala Karnofsky will talk about this a little bit later, trying to get to a more rapid way of understanding what's going on. You'll learn about normalization of data, especially what the untargeted platform from George Michaelides a little bit later this week. When you run a large number of samples, what you get is some variation from batch to batch and missing data. And through normalization processes, we can actually get a reasonable normalization and interpolation of the data, the missing data, such that you can have an ability to really get a good understanding of the broad metabolome of the samples that you're looking at. I'll show you one sort of surprising but actually very successful experiment that we did. This is with Dan Teitelbaum. The paper is actually sitting on my desk to read. I need to get back to him. But basically, what he's very interested, he's a pediatric surgeon. What he's really been interested in is why children with short gut syndrome who cannot absorb their food properly, when they're put on total parenteral nutrition, they actually get a high rate of infections, of infections in their blood. And the question becomes, why does this occur? And so Dan's been working with some gnotobiotic mice, and what he did is he enterally fed a control group. He did an enterally deprived total parental nutrition group, and then a gnotobiotic or a germ-free mouse model. And what we did is we took the effluent of the bowels in these animals and did untargeted metabolomic profiling. And what we found is that when we looked at the top hits, when we did the profiles of the effluent in the gut, is that it was all these amino acids, some unknown compounds, which actually some of these are probably amino acids as well. And then it actually occurred to us that this looked just like the solutions that was being infused into the animal. So the relative concentrations of the different metabolites that were in the gut by the untargeted platform actually turns out to be similar to the TPN solution itself. And so we had the idea that probably what was happening is that there was a breakdown of the wall, and instead of things being absorbed from the gut, the food being absorbed from the gut, in fact, it was going the reverse way. And it may be the breakdown of the gut barrier allowed the effluent of the plasma, or an ultrafiltrate of the plasma, to go into the gut. And the thought is that this effluent actually changes the flora of the bowel. And so by infusing nutrients in IV, that ultrafiltrate goes out in very high concentrations and then changes the microbiome in the gut, leading to inflammation and further breakdown of the gut. And so to test that, worked with Dan and infused C13 leucine into the animal at the same time, and then looked at the amount of leucine incorporated into protein in the gut. And indeed, there was a robust incorporation of leucine into the bacterial, the microbiome and the bacterial proteins, suggesting that this indeed was occurring and that the nutrients that we're infusing was actually being utilized. Show you just a real quick fluxomics experiment. We'll talk about in vivo and in vitro fluxomics a lot in the next couple of days. There's a paper that Charles Evans and a graduate student, Katie Overmeyer, published. The reason I like to show the cover is because my daughter drew it, and we were really proud. My wife and I were on the paper, and my daughter, drew the cover. It was actually a lot of fun. And so this was actually looking at the fuel utilization in skeletal muscle in vivo while animals are running. And so what Charles and Katie did was taking two types of animals, the high-capacity runners, these were animals bred for excellent running capacity, and low-capacity runners back here who don't run very well. And the bottom line is that the high-capacity runners utilize fatty acids as fuel much better than the low-capacity runners who use carbohydrate, and this seems to allow them to run farther. But also what we noted in the studies, and this came out of some of the bioinformatics work that was done, is that these animals also use branched chain amino acids, or apparently use branched chain amino acids as fuel better. And so what Charles and Katie did was, while these animals were running, infused C13 valine into the animals and looked at the accumulation of label in different metabolites through the branched chain amino acid catabolism pathway, and noted the marked increase in the use of the branched chain amino acids in the high-capacity runners. So not only do they utilize fatty acids more robustly, but they also use the branched chain amino acids more robustly. And what they found, this is related to the deacetylation of the proteins in the mitochondria that actually are responsible for branched chain amino acid utilization. So there seems to be not only an increase in the amount of the protein, but a marked deacetylation, allowing the flux to go through these pathways much more easily. And so I think this is a really nice example of the integration of different omic technology, the proteomic technology and the metabolomics, and giving you an integrative story of things that happen. Now, statistics are really one of the most important, one of the most sought after help that people come to our lab for. Once they get the metabolomics data, people always want to know, now what do I do with it? And what we're going to hope to tell you is that it's difficult. Some of this stuff is, I want to say this the right way. The targeted stuff, pretty straightforward. The untargeted stuff, when you get very large data sets, it's just like any large data set where you have missing data, which is inherent into the untargeted platform, that you need to be able to develop a systematic way to looking at the data. We have some capabilities, but the capabilities that we have far exceed the amount of requests. To Nu, I always worry that she's going to leave us, and Sasha as well, because people are continually hounding us about how they can actually look at their data. Dong Yu is here. Dong Yu, are you here? There you are. Dong Yu, she's actually been working for the core. She's a biostatistician. She's been working for the core for about an hour now, right? So Dong Yu is actually a new biostats person we hired specifically because the amount of data and statistical analysis that we need to do to help the core out. So We're going to have a number of lectures by George Michaelides and some other people really, giving you at least the primer about how to utilize this data. One of the things I would urge you to do if you're going to do untargeted metabolomics data is find yourself a good friend who's a biostatistician, because it's going to take some work to get this done. Brian Mickey knows all about this. Anyways, so here's some statistics about statistics, and I won't go through any of that because, frankly, I rely on people like Tanu and Sasha and George to get this kind of stuff done. Now, we've been developing, one of the things that we really have been trying to do, and you'll hear about this later in the week from Alla Karnowski. Alla developed a really nice platform for looking at metabolomics data. And it's based on a Cytoscape backbone. So I don't know if anybody's used Cytoscape for gene expression profiling, but she's developed a plugin that can be integrated with gene expression data, used by itself or integrated with gene expression data, and it can do time courses and things like that, that will be able to give you pathways that your metabolites are occupying. And so she'll go through and tell you exactly how to use this, but it's really been a great tool for both myself and for a lot of people outside the university. We're trying to develop other tools that are associated with that. Alla will tell you a little bit more about how we're trying to develop ways of using statistical analysis of relationships between known and unknown metabolites to be able to identify these unknown metabolites, or at least place them in a network such that you can see how they respond to different biological perturbations. And so this is an example of a paper that hopefully will get out soon, but I just sent Alla an email about how we need a little bit of work. So basically, this just takes correlation coefficients between these metabolites, but then does partial correlations to try and improve the closeness and the intimacy of the relationships, such that you get a much higher statistical association between these metabolites. One of the things that this has helped us do is finding things that are sort of, quote, junk. Sometimes molecules will break down the source of the mass spectrometer, and you get a peak, and it's a very reproducible peak that's found, but it really just represents a fragment of another compound. And so it gives you more data, and when you're trying to do your correction for your Bonferroni correction, when you have large numbers of data, at least we can collapse the amount of the world that you have to look at if you're going to do these kind of things. So Alla will go through that in more detail over the next period of time. Or next couple days. This Metascape platform is actually, it looks like a mess, but this is really very cool data. This is some of the stuff that we used when we did this paper about the rats that can run, and this is integrating metabolomic data and transcriptomic data. And the differentially expressed ones are the metabolites are blue, and differentially expressed genes are in the red, and the pink ones are fillers of the pinks are fillers of genes that are in the pathway. And so what you can see that you can integrate these things, and what this program does, it will give you an enrichment score and tell you which pathways are more markedly enriched in your samples, such that you can zoom in and actually pull out. And I didn't put a picture in here, but if you click on one of these, or a picture in here, if you click on one of these pathways, it'll actually extract just a subset of this pathway, so you can actually explore it in much more detail. So I would urge you to go and listen to Alla's see Alla's demonstration. It's really a very useful tool. And just an example of some of the untargeted metabolomics, this is a paper from Scott McDonald published about a year and a half ago now, where working with Sasha Raskin in the core, did untargeted metabolomic profiling and found differences in expression in a large number of metabolites when they went through and used some programs to look at the enrichment of the different pathways. What they found is the glycolysis and purine metabolism pathways were markedly enriched in cells that had an overexpression of the ALK oncogene. And what they found through this is that the pyruvate flux was markedly increased, which led them to them investigating PKM2, which is one of the rate-limiting steps in the glycolytic pathway, which is controlled in cancer through oligomerization of the specific subtype of PKM. And so what they were able to do was actually understand the pathway of the increase in flux through the pathway simply by doing the untargeted metabolomic profiling, then going back and doing targeted metabolomic profiling and putting it together with some proteomics. And again, it was a nice example of the integration that you can do once you do the untargeted platforms and integrating with the other data that you have. You can make you can gain some biological insights that you might not otherwise have. So I think I'm going to stop there. They have no easy clock in this room. To tell you, see if there's any questions. So basically, what I want to do is just give you an overview of kinds of things that we're going to talk about, some of the things that we've done. What we're going to do is intermittently, as Barbara said, that we're going to try and give some cases because, seeing some examples how these things are used can be very useful for individuals. So I'll stop there and entertain any questions that people have. Yeah, wasn't high content. All right.
Do we have access to all the slides? Or?
Yeah, I think they're all Terry.
Yeah, they're in that box, and if you have trouble accessing it.
Yeah, so you should all have gotten. The internal people know how to use Mbox. The external people have access to it as well. Yeah, so all the slides should be there. We just wanted to save trees, and we don't want to print them all out for you. If there's anything else that you want that's related to this, I'm happy. We have lots of slides laying around. So if it's published, we're more than happy to give you everything. Okay. Yes, ma'am.
So really, after you guys identify the metabolites, do you guys run other kind of experiment to validate your discovery?
So we do what you like us to do. So we try and be a full service core. So if you identify metabolites of interest, either knowns, and we can do it with unknowns as well, we can set up targeted metabolomics data or methodology so you can see in a quantitative manner the relative levels of these metabolites in your samples. So absolutely. So that's exactly the kind of way, with the last experiment, that's exactly what we did. We found metabolites, set up targeted metabolomics data so you can actually quantify things.
So visually, mass spectrometric method.
Yes, yes. So that's, like I said, that's mostly what we do. If you do find that NMR is necessary for either structural identification or you want to do your assays for some reason, non-destructive assay of your samples, we can get you to an NMR machine if that's going to work for you. So maybe as a corollary of your question, if there's an unknown metabolite, doing structural identification, we will help you with that. Sometimes it'll take more time. Hopefully, with some of the technologies, the informatics technologies that we're developing, that will be able to narrow the world of what this specific metabolite might be. And with some of the other platforms that we've developed, with the lipidomics platform, because a lot of these unknowns are probably lipids, hopefully we'll be able to help you get there as fast as you can. But sometimes it's a little bit tougher. Les?
So can you say something about like trying to do an investigator comes to the core and wants them to do work with them to try to do a little pilot study to make sure there's something to study?
Yeah. So in other words, what's it going to cost you, or how do we go about doing it, or? So, I think Maureen's going to talk about interacting with the core. But in general, this happens all the time. Somebody wants to come and they want to look at, glycolytic metabolites. What we usually do is it's either myself or Sub will, or Maureen, or people with content expertise like TM we'll discuss with the individual exactly what you want to do, and I think from the experience that we have, we'll make some recommendations. So if you want to do tissue culture experiments, usually if it's some cell line that we've worked with, we can give you some ideas of initial conditions that you may want to use to start out looking at the time course of your experiments, especially if you want to do fluxomics studies. It's really going to be important to really get the timing down correctly. If you want to look for specific metabolites in specific samples, we can set up assays to do that. We've done that for individuals. We tend not to take work for looking at drugs and drug metabolites. If it's part of another study, we have made exceptions, but in general, we refer drugs and drug metabolites to colleagues in pharmacology. It's just we don't want to become a drug metabolism lab. We have enough business to keep us busy. Does that sort of answer your question? So it's really, and actually I should say this, and I think Sass will, when she talks about sample preparation, will emphasize that it's really important that before you submit samples to us, and I'll show you this afternoon some vignettes of things that you shouldn't do, but it's really important to talk to us before you come to before you submit samples, even before you do the experiment, unless you're taking the archival samples out of the freezer, to really be able to do this, to be able for us to help you make sure that we don't waste money, because it really is. Because if you submit samples, and people have gotten upset with us, they've submitted samples and then it's sort of like it didn't work really well, and sometimes we can't be responsible for errors that people do when they haven't discussed it with us. We have to charge you for it. Right. I want to talk to them too, because they're going to be talking about their experience working with the core. So that's good. Yeah. You won't hear that. Yeah. I spend a lot of my time talking to people, and we've been turning away a lot more people because now that we've gotten more experience, sometimes when people come to us with specific kinds of experiments, it's not going to work. We just don't want people to come to the core and just run samples and waste their money, and so we'll just turn people away. They're not happy about that either. I'm not sure why. But anybody else? Okay, I'll stop there. Next on the thing. Oop, I didn't bring mine down.